All right, Breasties. A nice warm introduction for Dr. Adrian Lee with What Now? The State of Metastatic Breast Cancer Research.
I like that. You see, Isaac said he was going to be the warm-up for me, so I thought that was good. He was going to get you guys excited. And so I am going to be telling you about metastatic breast cancer research. I only have 10 slides, so I've kept it slide light, so I want dialogue. If you don't say something, you don't ask questions, this is going to be really short. So you can ask. There are no dumb questions, there are no baby questions, there are no naive questions. You can ask whatever you want. Yeah, that's good. So first off, myself, as you can tell, I'm not from the United States. Actually, I was born in England. My mother had 5 boys. I am a twin. I have a twin brother. She actually was diagnosed with endometriosis when she was younger and was told she couldn't have children. And then she had children pretty late, her first boy, and then had another, and then had a third boy and said, oh, she wants a girl, and she had twin boys. I'm one of those two, so I'm the black sheep of the family. She had breast cancer. She was diagnosed with breast cancer over 20 years ago now, with early stage ER-positive breast cancer, got phenomenal treatment. We have phenomenal drugs for that, and is now 94 and doing fantastic and extremely well. Thank you. So I left the US, England, to come to the US, which is, one of the great places to do research. It's kind of struggling at the moment, and I'll have a little bit of a discussion about that. Not too much, but a little bit. But I came to the US. I came actually to San Antonio. Have any of you been to the San Antonio Breast Cancer Symposium? Someone there. If you get a chance to go to it, one, it's a great city. It's a fantastic city for vacation. It has this amazing riverwalk in it. It was the epicenter and how breast cancer research started in the US, and that's why it hosts the Breast Cancer Symposium. So I went there because it was the best place to do breast cancer research, and I met my wife there, Steffi Oostenrijk. Some of you may have met her. She was here a couple of years ago. We share a lab together, and we do research together. So I was lucky to meet her there. Actually, that was the place, Isaac showed you a slide about the discovery of HER2. And HER2 was discovered by a guy, Dennis Slayman, who's in California. And actually, the senior author was Bill McGuire. He was in San Antonio, and it's called the Will McGuire San Antonio Breast Cancer Symposium. So basically, it was a guy with amazing foresight to take these breast cancers, build this cohort that led to the discovery of HER2, and that's why it became the epicenter for breast cancer research, and that's why everything kind of started there, and that's what brought me there. So I had, with Steffi, we have 2 daughters who were born in San Antonio. They're now kind of moved on. I'll show you a picture of those. And Steffi and I have been doing breast cancer research now for about 30 years and thoroughly enjoyed it, thoroughly coming to these events, speaking to lay audiences. You are the reason we do it. This is why we do our work. And you are a major player in the research that we do. All of the research we do is on patient tissue or patient blood or something to do with patients. And if you don't participate in that and don't volunteer in that, then we don't have anything to do, yeah? And every advance we've made has come from you giving forward to the next generation. So it's incredibly important that you participate in clinical trials and continue to do everything, all the great things you're doing. So I have 2 disclosures. The first one is I have a PhD. That means I am a doctor of philosophy, yeah? Just be clear on that. Our daughters, who are here, like to point out that Steffi and I are not real doctors. You just heard from Isaac, who is a real doctor. So if you need medical advice, don't come to me, because it'll be terrible. Go to Isaac, and he'll give you really good. He can tell you about medical oncology. I would just say this is Nicola, the first one. She just graduated from law school and is a lawyer now. Paula, the one in the middle, lives in Austin, Texas. She works in pediatric hematology oncology. And this photo is kind of funny. I won't go into it now, but this is a couple of weeks ago, 2:30 a.m. in Mexico City at a wedding. It was the most amazing event. But if you want to know more, I can say outside of this, all the crazy things that happened that evening. My second disclosure, which sadly we're in an era where I wouldn't normally make this disclosure, but I have to make it now, is that the opinions I express are my own and do not represent the institution I work for or the funding agencies that I get money from. So in this era, in this administration, there's been a lot of challenges that we're facing, and words can be taken out of context, they can be presented out of context, and our institutions are telling us not to talk to the media. And if you hear any kind of verbiage from people at institutions, they'll always start with, "This is my personal opinion, not of the institution." And it's the institutions shielding themselves from people speaking out of line and then them getting targeted, which I think you've seen that many times now. So it is a challenge. I said on this, please do not record or post what I present. We're at the Breasties where they put everything online immediately, so I have a sense that's probably not going to happen. But I will try to be careful with what I say, and maybe you can try and be careful with what you say as well. So when I was talking with Bree and Kelsey about what I was going to say, I wanted to bring across to you some of the advances we've had in the treatment for metastatic breast cancer. Because I think you realize in the news, you nearly always hear these negative things. It's not really very inspiring when you watch the news nowadays. There are some inspiring stories about new treatments, new advances, new diagnostics, ctDNA. You're hearing that here constantly, yeah? These are relatively new things that are extremely exciting and are probably going to transform how we treat and manage breast cancer. But they sadly come at a time when we have a lot of kind of bad feelings about scientists. You know, a lot of this started with COVID, where scientists are bad, they're not telling us the whole story, vaccines are bad, et cetera, et cetera. And it's really come to this, what is now referred to as this existential crisis, in that there's been this huge, as you know, cutting of funding in the federal government. I think most people might agree that there's probably bureaucracies there and poor spending of money in the federal government. And the idea was to basically take a kind of chainsaw to it and just chop pieces to bits. And a lot got cut out that probably shouldn't have been cut out. And a lot of that happened in the medical space. And so some of this, and I heard a lot of people here earlier in the meeting, and then also when I talked with Bree and Kelsey about that, oh yeah, I've heard this on the news, but I didn't know if it was directly affecting you. It's absolutely directly affecting us. It's bad in that it lowers the morale for the people in the lab. This isn't an easy job to do, being a scientist and being those lab personnel. They work long hours. They have a very strong passion, as Isaac said when he was going up on the vomit comet. They have a passion for what they do, but a passion can only go so far, yeah? You need to have a white light at the end that says, maybe there's a career here for me. If there isn't training and training money for them, these people are going to kind of go away, yeah? And so what we've seen in the last 3 months are some of these cuts are having massive effects on our industry and our biomedical research, such that, for example, I'm from Europe, many of the European institutes are trying to now recruit Americans back to Europe. So it used to be a brain. I'm a classic example of a brain drain from Europe to the US. And there will now be a reverse brain drain. And it's already started with scientists picking up from the US saying, hey, probably the glory days are gone. I'm going to go back and do my stuff in Europe. So this is a major issue. And what is happening, is starting to happen, may happen, is you could, for example, get delays of clinical trials. A number of clinical trials, as you've heard, have been shut down right in the process. Some of them have had reduced funding. This probably is going to affect many of you, or could affect many of you. We've had limited access to treatments and obviously reduced research opportunities. As we don't innovate and we don't generate these new ideas, as Isaac was saying, it's these ideas that happen in the lab that then get translated into patients. I'd be happy to take any questions on this. I know I've been asked by several of them, how it's affecting you. As I said, we haven't lost direct funding in our lab, but our institute has lost funding and others have lost funding. We lost funding for our training program, for example, for underrepresented minorities. Yeah. Yeah, to try and, yes. Yeah. And I heard this question. I was called to the back, and I heard this question was asked to Isaac, and I didn't get to hear his answer, but I think the similar question was, what can be done? I think, and I often am a little bit despair about this, is my voice going to have an effect in this space? But I do think breast cancer is pretty unique in that it's really common. And so the reason why we have funding from the Army, for example, from the DoD, is because that's where a lot of breast cancer advocacy started, was because it affects a lot of the, spouses of, veterans, for example. And so suddenly money came towards it. So I think definitely going to your congressman, statesman, just going to those officials and pointing out these problems. I think they listen to their constituents. And if enough people speak up about it, which they are, then you can do it. There are these science advocacy days, just general ones. There's lots of movements on the Hill at the moment to try and stop a lot of this. I mean, if the bill goes through for the Medicaid, that's going to affect obviously millions of people, yeah. So I think advocacy is one of the only things we can do at the moment. But I think we need advocacy. We certainly need advocacy from patients. Yeah, you are the ones that have been through this. In the need for research, in the need for clinical trials, you have the biggest voice of anyone, yeah? And so we rely on that to a large extent. Yeah, and first.
As normal, and there's new developments there. Do you think that we will be able to find a way to get access to?
Yeah, to benefit from it, yeah. I heard, so the question is, was that me? Is that someone saying, don't speak? Is that someone saying, don't speak so loud?
No, that was my bad. I said checking the mic.
Okay.
My apologies. Love you. Bye.
Coming from somewhere. Yeah, okay. That scared me. I already thought someone's listening in and telling me not to answer that question. Big Brother was listening. Very good question. I was asked this earlier. So the question is, if the research moves over to Europe, Asia, etc., will we still have access to that? It's a very good question. One would think so, in that we often get asked by patients, do we share our research knowledge? Are we sharing this? And we're like dumbfounded and say, yeah, of course, this is like how we work. One of the most eager things we do is to tell others what we've done, because it accelerates them and it makes us better. So we share all the time. We have to be careful about sharing, because there's some competition between us. I compete with Isaac, and I don't tell him everything, but and he's naive and he tells me everything. So I think it will, but one can imagine, for example, if much of the research moves to China, which will probably be the biggest powerhouse coming forward, that may not be shared back, and so we will lose. We will also likely lose the innovation. I mean, that's really key. America has been the research innovator for a long time, and a lot of that has then gone over to Europe, and they've benefited from it. It's kind of guesswork as to whether we would, reverse benefit from that. But I think that's a great question, and yeah, not a real good answer. We'd rather not be in that position and have to guess, yeah. The next question, yeah. Very good. With a sense of humor.
I don't know if this is a separate question, but should we be worried about FDA approvals slowing down?
Most definitely. So those cuts from the departments were across every department. So the FDA took cuts, the CDC took cuts. We don't know. Obviously, there's immediately, throw my hands up in the air in despair. We don't really know how this is going to settle, but it for the moment, it obviously doesn't look good. That those cuts are going to hurt multiple departments. There is something to be said for efficiencies, and I think we all think government's too large, and maybe cutting government a bit will be good. But some of those key groups got cut, and obviously there's some rehiring now of them, like weather forecasting, for example. It'd be good to know if we have a hurricane coming through here, yeah? Well, we need people to forecast that, yeah? We need FAA, we need people to track our flights. That's become a problem. So I think it's pretty clear that some of this is going to kind of level out over time, and has to. Yeah.
And this is no way to out you out to give us the real tea.
Yeah.
But
that's good.
There's so many organizations like the Cancer Society that does all of this fundraising and things like that, right?
Yeah.
And then it says cancer research. Are you guys receiving these funds to keep it up? And if not, let us know, because we can boycott them and then really figure out where the money is going, right? So I feel like in
I
this,
see why
in
you
this-
started that with that. You're not going to ask me directly.
and you don't have to be the one to tell us that they're not, receiving the funding,
Yeah,
but
yeah.
it just seems like, this is the time more than ever to give the funds that are being raised to save these clinical trials.
Yeah, so I'll answer that in a general sense. It's a great question. We are going to have if this all finishes as it started, we will have a change in the funding, the type of funding that funds biomedical research in the United States. I mean, what's changing is, should the government be funding all of this, or should someone else be funding it? Should the pharmaceutical industry be funding it and not the government? That's a fundamentally interesting question, yeah? Yeah, we can all have our own opinion and we can all have discussion over that, but it does change the way we're going to do this. And so there's quickly been this question of, we're very dependent on federal funding. Let's diversify how we get our funding. Let's try and get more from, and one of the ones has been the foundations. There's going to be this incredible pressure upon the foundations to step up with their funding, and this is going to be a problem for them. There's no way they're going to compete with what the government or substitute for what the government was giving us. So I do think some of those cancer societies are incredibly important for what they're doing in the policy space. That's like, as we said earlier, about advocacy. It's incredibly important to advocate and go to the Hill and kind of pressure these points. But making sure, I mean, I work for foundations where nearly all of the money goes for research. I mean, the Breast Cancer Research Foundation, nearly all of it goes for research. It's known for that. Yeah, it's like a A-plus foundation. So I think there will be a changing as we look harder into how money comes into research. There will be a changing of this, and it'll be more apparent that we need more to come to research to kind of fill the gap.
Okay, so yes and no, ladies, hold off on the boycott. I'll
I
let you know.
I would say let's hold off on the boycott for the moment until we see how this lands. I like that answer. That was very kind of you. Any other questions? Yeah, right at the back.
I'm coming.
We have someone running to you.
Literally running.
Last question, and then we'll move on. I wanted to keep this short because I want to get to the better stuff, the advances in breast cancer treatment.
Oh, thank God. I showed up a little bit late and I thought you were done and I was like, okay. Hi, everybody. My name is Katie and now I've completely forgotten my question. So as we're seeing this shift from, from government funding and then the private funding is kind of moving in and my oncologist also kind of encouraged me that like, hey, not everything is government funded. Yep. What are the things, and because you've got a lot of pretty serious advocates and activists here,
Yep.
like you tell us to go and we'll do it.
Yep.
What are some things that we can be doing to help support the private funding and the organizations that are trying to continue this type of research?
Yeah, no, and we appreciate that. And I think it's a really great question. And one of the things is really spreading the word of realizing this is having an immediate impact upon us as researchers. Yeah, I was asked multiple times, well, I've heard it on the news, I'm not really clear. It is happening, and it's changing our research infrastructure. Like I said, one of our major training grants just got stopped. And this is apparent across the board. All universities have had this. So I think you have this amazing influencer presence here. I'm not sure I understand influencing yet, but still, you have this presence. You can spread that message, that this is having direct impact on breast cancer research that will ultimately affect breast cancer treatments, This all comes from basic research. All of that funded research is what led to every advance we've had has come from research. Every advance. None of them were just like, came out of the blue, someone researched it. Isaac went through these kind of the scientific logic that leads to these things, So this isn't just luck that we got to these places. Someone took time to research it and figure it out. And you need to spread that message. This is the most important. It's kind of the saddest point that we're at this amazing, as I'm going to tell you, inflection point of advances in the treatment of metastatic breast cancer, and we have the least amount of money to do it now. And that is really kind of sad. So let me now, having said that, move. So I wanted to get us off of the sadness of the current situation. And Sam, he actually unfortunately passed away, but he passed at the... I know. At the wonderful age of 14, of old age. But every day, He was happy every day. He had love. It was like I'd never seen him. Every single day it was, it's a new person. It was crazy. So, but I wanted to highlight just one slide on each, 4 major advances that are coming in metastatic breast cancer that's going to improve outcomes for patients. Some of them you've heard multiple times at this meeting, and it's not surprising because these changes are coming so fast and hard at the moment, and they're causing a great excitement that a lot of them are coming. They only happened a week ago, and so several of you have asked questions about them this morning, actually. So some of this is going to be quite repetitive. This was my ideas, but you've already discussed many of them. But I'm going to say them again. So the first one, you've heard this a lot, and maybe we could take a bit of time on this to explain it, because you've heard this word come up, Serena 6, many times. So maybe I can just step back and just explain what this thing is and why everyone's so excited about it, and why it's going to fundamentally change how we diagnose, how we treat, and how we manage breast cancer. So there's this thing called a liquid biopsy, which is measuring circulated tumor DNA. So it's called liquid biopsy because normally we would biopsy your tissue. That's called a solid tumor biopsy, and that could be your breast, or it could be in your liver, or it could be in your lung. That's invasive and quite challenging and quite painful and quite difficult. The liquid biopsy is just taking it from blood. It's just liquid, yeah? And pretty much anyone can give blood. I gave blood last week. And this actually is an interesting field, and it didn't start in oncology. And Natera didn't start in oncology. They actually started in OBGYN. So their original test was non-invasive prenatal screening, or non-invasive prenatal testing. So when a woman is pregnant, we want to test to see whether the fetus has genetic abnormalities. Yeah, this is common, it's been done for many years. And that is generally done with amniocentesis, where you take this really long needle, you stick it in there, has quite high complication rates, highly invasive and quite complicated. And what someone found was that if you take the blood, I'm sure many of you have been pregnant and had children, your blood at that time when you were pregnant, if there's DNA floating in your blood, 10% of that DNA is from the fetus. It's shedding blood and going into your blood. 10% of all the DNA that's floating around in your body is from the fetus. And so someone had this great idea, hey, why don't I just take that and I can look at the genetics of the fetus just in the blood, rather than sticking this amniocentesis directly into the placenta. Turns out that worked, and that was the derivation of the NIPS tests, and if you have daughters, they're going to have that. And that's now transformed how we look at the genetics of a fetus. Is that all clear? Anyone didn't understand that? Okay, that's how it started. Now, someone then had the idea, oh, well, if that's true of the fetus, wouldn't that be true of the tumor? Maybe a tumor, when it's in the breast or the liver or the lung, dies off and sheds itself and ends up in the blood. It's got to get out of your body somehow. It's got to get out through the blood. So why don't we take the blood? And it turns out exactly the same. So basically, when you have a tumor, it is losing features of it into the blood. You can draw the blood, and we can find that in the laboratory using sophisticated assays. And this is incredibly easy to do. We do this on every patient all the time. And so this has, the beauty of it is you can do this multiple times. You know, you can only do a biopsy on your breast or your lung or liver once or twice, maybe. I'm sure you're not going to want to have it done 5 or 6 times, yeah? You can take blood every month, yeah? Meaning we can monitor it. So this is going to transform what we do in that, one, it's going to be used for early detection. It will be used just like we use mammography screening, which has been great, but has, you know, kind of low sensitivity. It will be used for early detection of breast cancer. In probably 5 years, we'll all be having a blood test that tells whether we have breast cancer or not. It will also test whether we have prostate cancer or any other type of 50 types of cancers. So you can use it for early detection. You can use it for understanding the disease you have. So just as we sequence your tumor, you can just look in the blood and see what type of tumor you have. And you could look at the aggressive nature, what types of treatment we should give. And then you can monitor it over time, like the Signatera test does, that says, okay, I'm just going to measure how well the treatment has done. Has it gone down over time, for example? So this is happened all within the last few years, and there's incredible excitement because these tests, like the Signatera test, is made specifically for you and you only. It's the ultimate precision personalized test. They take your tumor. Every tumor that you've ever had is all different. Yeah, it's like a fingerprint. No 2 tumors are the same at the genetic level. They may be the same in terms of you know, what Isaac said about estrogen receptor positive versus HER2 positive versus triple negative, but ultimately every single one has a unique fingerprint. They identify the fingerprint, just like you would on a, you know, for your clear or whatever. They identify the fingerprint and then measure that fingerprint in you. And it doesn't work in anyone else. It only works for you, in you. So it's pretty amazing, yeah? We make a test just to monitor for you. And that's an amazing advance. Any questions on that? Yeah.
Tumor or a progression, does it make sense to have this type of test? So if you have shown no evidence of disease for years, is it worth doing something like this?
Yeah, this is, and I should say, not surprisingly, with the excitement of a new technology, there's more we don't know than we know. And you heard that on the panel actually this morning, yeah, when we were asked about, well, why is my physician saying I shouldn't be tested? Because the physician doesn't really know what to do with the test, because a lot of this stuff we just don't know. We're incredibly excited and we know it will be transformative, but we need to build the evidence base, and that takes time. And it's a sad thing to say, but it really does unfortunately take time. But I would say, for example, there was a study done in patients with ER-positive breast cancer. They had surgery. They were given anti-endocrine therapy, and they were tested 5 years later. So they've been disease-free for 5 years. And they said, well, how many actually have any at that point? So at that point, I think like 8% were positive, and several of them then went on to have recurrent disease. So we need to do those studies to figure those questions out. Like you said, you know, when, this is kind of what was said on the panel, you know, when should we be testing? How often should we be testing? You know, how are we testing? What tests are we using? Those are all questions we need to know. And they're exciting, and we hope you all participate in that. You know, that's the one thing you can do, is participate in research, participate in a clinical trial, donate your blood. This is how we will learn, and this is how we will move forward. Yeah.
So I know probably a lot of us have used like the Signatera test.
How many,
like
hands up, how many have used it? I'd be interested to know. Wow, that's crazy. Yeah, so 30 to 40%, great.
So my question is, like, at least with Signatera, it looks at our primary site tumor, and as cancer evolves and we start to have other organs impacted and things like that when we're metastatic, obviously our tumor DNA can change and morph over time. So, you know, with Signatera, it just tests at that original level. Like, are there things being done in research now to look at the evolution of the tumor and being able to pick up those kinds of things?
So look at this, a highly educated question. That's a really state-of-the-art question, which is really great, really excellent.
Thank you. What up?
And luckily, I think I can answer it as well.
Okay, good.
So I think Isaac mentioned this earlier. A major challenge in the treatment of metastatic breast cancer is that it evolves. So it becomes drug resistant, it changes itself. It can even change the subtype. We've published a lot on this, and others have, that it can go from luminal B to HER2. So it's adapting, and it's pretty clever, yeah? It's so kind of facile, that's why it's evolved, and that it says, okay, take away this chair. Isaac said that. Take away that chair, it'll go and find another chair. And you can, you know, it's like whack-a-mole, you can find multiple chairs. So it's evolving, and we need to understand that. And we couldn't measure the evolving, how it evolves, because we can't keep biopsying. With the blood, we can do that. But that's very important what you said, in that you need tests that can find these new markers. The Signatera doesn't do that, yeah?
Right.
It only measures the markers you had at the start.
Exactly.
It can't look at the evolution. But of course, they're thinking about this, everyone else is. And so there are tests, new tests coming online. I feel sure Natera will have one as well, soon as well, where they actually then look for these new changes.
So
and I'll tell you about...
soon, like in the evolution of like research to FDA approval to market, like obviously
Very
there's
soon.
time. Okay.
Very
so
soon. One's already out there. Natera will be out soon. Yes. No, they will.
Okay.
These are coming so fast. Yeah. So I was about to say within a year. I was going to say within a year. Yeah. You will see. These are happening in real time as we speak. It's incredibly exciting and obviously so transformative. So this is the New York Times from 2 days ago, June 1, well, 5 days ago now, reporting from ASCO. So something at ASCO that makes it to the New York Times, it's going to be pretty important, yeah? And this is the SERENA-6 trial, which is going to transform what we do. And as you heard, so basically in that trial, women were on a drug treating their ER-positive breast cancer, and they had blood taken I think every month or 3 months. And they're looking whether it evolves or changes. So does it get mutation, does it gain a mutation in the estrogen receptor, also called ESR1? That's important because that's the receptor that's targeted by the therapy you're giving. And what the tumor is doing, the tumor is saying, hey, you've turned off this chair, I'm going to go and get another one by mutating the target, yeah? And we can measure that in blood. And we can measure it really well in blood. And what that trial said was that if you monitor this while they're on therapy and it appears, they then randomize them to either keep on the same line of therapy or switch over to a different therapy, with the idea that maybe switching to a different therapy will help them because the tumor has evolved.
Is that the same with the PIK3CA mutations as well?
So similar with PIK3CA mutations, yeah. And so that arm, where they switched therapy, they did better than on the continuous arm. That's the first example of basically switching a therapy based on a molecular phenotype and not a clinical progression. And that's why it will change, because this was done earlier than clinical progression, and there's a lot of excitement. Now, as Isaac said, his kind words were, there's lots of nuances to that trial, yeah? People spend thousands of hours designing these trials, the patients go on them, they try to do their best, and the moment it comes out, there's more questions than it answered. And that's good, yeah? That's why we do research, yeah? You know, it's kind of frustrating. I'm sure to the people in the trial and the people who ran the trial, it's frustrating. But ultimately, the trial was positive. It's a good trial. It'll lead to better types of treatments. And we can imagine lots of other scenarios where we would do something similar. And so there'll be lots of kind of follow-on from that trial. Any questions about that? Okay. So this, as I said, liquid biopsies is going to be transformative to how we move forward. So you heard about ASCO. This happened last week. It's the American Society for Clinical Oncology. It's the major 40,000 people go there and present the hottest research. And breast cancer was the leading cancer there because it had multiple advances that will change guidelines for how breast cancer is treated. And I put this up there because I thought these are the four major, everyone may have different kind of feelings on what was major. I put these up here because the nice thing was, you see all of the subtypes of breast cancer were represented in the advances. So we have, this is nice inclusion, yeah, that there are advances. Everyone always says, hey, you're not studying I hear this all the time, you're not studying my subtype. But there's advances coming in all of them. So the first one is triple negative breast cancer. This sacituzumab govitecan is one of these ADCs, or antibody drug conjugates, that Isaac talked about. These are these smart drugs that are targeted to the tumor with chemotherapy. This is kind of what we always wanted. Why do we give chemotherapy all over the body and cause this toxicity? Why don't we just give it directly to the tumor? That's what they do, and that's why they're so successful. And they, in the last 5 years, are really transforming how we treat breast cancer. So that plus pembrolizumab is an immunotherapy like he was talking about. This will likely become the new standard of care for PD-L1 positive triple negative breast cancer. So that's always exciting for physicians when they see a good result and they think, yeah, okay, I now have an improved arsenal, and they'll have those other drugs they can move to. For the PI3 kinase mutant, this is the one you just mentioned, the PI3 mutant breast cancer. There's a trial with, I should say, sacituzumab govitecan was one of the first drugs to show overall survival improvement in triple negative breast cancer, which is hard to show, and so that's why it's been so popular. This trial they always have names you can't pronounce. They do this for various reasons. That's another story, but inafaslib, palbociclib, fulvestrant together improved overall survival in this phase 3 trial. This, again, will probably change treatment, and again, changing overall survival is what we want to do. We don't just want to slow the progression of the disease. We want to improve patients surviving. In the estrogen receptor positive disease, that's HER2 negative, we have vepdegestrant, again, a name you can't pronounce. You're not supposed to pronounce that name, which significantly improved progression-free survival for ESR1 mutant. That's what we talked about in SERENA-6. That has a specific mutation in estrogen receptor. So this is, we've been targeting estrogen receptor for 30 or 40 years, and we still have new drugs coming out that are doing better than those drugs. And this is important. Yeah, we don't want to say, okay, we've reached enough, let's stop doing it. They're still kind of innovating and developing new therapies in that space. And finally, HER2 positive, which is many of you know that we've made the most dramatic advances in that disease over the last 10 years. Destiny Breast 09 will be transformative. So that's Enhertu, which I'm sure some of you, all of you have probably heard of, and some of you have probably taken. So Enhertu plus pertuzumab in HER2-positive breast cancer. This was super important, caused quite a lot of controversy over whether how patients are going to take this and whether they can tolerate it and whether there should be a kind of run-in to this therapy. But this was really, physicians come away and they're, oh my gosh, they go into the clinic next week and suddenly patients are going to be asking for all these new lines of therapy. So super exciting, super important. And remember, as I said, this all comes from research, yeah? All of this isn't by chance. This is by scientists in the lab doing research, figuring out these new therapies, either in the laboratory or in pharmaceutical industry, and then translating that over to physicians who do clinical trials, who are funded through the Clinical Trials Network, which is federal funding as well, in concert with the pharmaceutical industry. And then this ultimately, we hope, will improve survival for our patients. Yeah. Okay, yeah. So this one here that you're not supposed, no, it's actually very different actually. So targeting the estrogen receptor has been the mainstay for ER-positive breast cancer for years. The first one was tamoxifen, which antagonizes it. It binds to the receptor and stops it working. Then we had aromatase inhibitors. So aromatase inhibitors lower circulating estrogen, the ligand, and stop turning it on. Then we have SERDs, fulvestrant, and now oral SERDs, camizestrant. They target it for degradation. This actually is something called a PROTAC. This is the fourth line. So these, I'm telling you the major classes. This will be the fourth class, which is a PROTAC, which is very novel, which basically kind of recruits to the protein a degrader and then causes it for degradation. So it gets much more complete. It kind of, remember how we're going to take that chair away? We're just going to obliterate that chair. Rather than just like taking it away, we're just going to like destroy it. That's kind of the equivalent. It takes the estrogen receptor and totally destroys it. The idea is we're already doing pretty good at targeting the estrogen receptor, so the changes are, I would say, somewhat incremental. They're not like you're getting with these other ones. But giving more options is always good, yeah. I mean, as you know, it's evolving, and so we want to have lots of different types of therapy that we can then use in different settings. Yeah.
I'm coming with a mic so everybody can hear your question.
And we'll talk to the last one, then we'll move on next to, because I do want to get to the end.
Can you briefly explain the difference between Enhertu and Herceptin? Because on this last bottom one, there's a little confusion for me on the difference.
So Herceptin, as many of you know, that was the first drug. It's otherwise called trastuzumab. So it's Herceptin or trastuzumab, and that was the original drug. It's an antibody or a protein that binds HER2 and stops it working. So it physically sits on top of it, like sitting on top of the chair and then stopping anyone else from sitting on it. It blocks anything from happening. And HER2 is taking that reagent that binds HER2 and putting a chemotherapy on it. So it's attached the chemotherapy to this protein, and then it finds HER2 and then gets internalized and delivers it's called a payload, a toxic payload, and it delivers the payload into the cell. So targeted. So HER2 is quite specifically expressed in the breast for unknown reasons, also in the heart, unfortunately, but very little expression elsewhere. So that's why you get this nice targeting and therapy kind of specifically in your breast cancer, particularly if it's HER2 high. So Isaac touched on this, but one of the things that I'm excited about, and this because I do this directly in the lab, is that we have new and better models and tools to study breast cancer. So I'll give you an example. If you take a bowl of Cheerios, which are all different types of colors, if you've seen the US Cheerios, you may have seen this on the news recently, the US Cheerios look very different to the European Cheerios because you have food dyes in the US that are illegal in Europe because they're toxic, and they're trying to take them out at the moment, unfortunately. But anyway, that's a side story. That bowl is very colorful, yeah? And each Cheerio is a different color. Imagine that's a different cell within your tumor, all those colors, yeah? The tumor has this kind of what we call heterogeneous multiple cells in there. For the last 30 years, our research has been to smash that bowl up into a powder, so it'll be this kind of yellow powder, and then try and figure out what the Cheerios were before. kind of with computers. We kind of like figured that out. And we did that because that's all we could do. Now we have technologies to dissect those individual cells, to not crush it up. And so we are getting a resolution of cancer and breast cancer specifically that we didn't have before. It's the equivalent. If you imagine how medicine was before the microscope, there was a lot of thing about there's this black bile that causes this. We didn't know about bacteria. We didn't know about anything. Now, with the microscope, we know that. This is similar. We have technologies that for the last year are telling us features about breast cancer that we had no idea about. Those technologies are unbelievably expensive. So we have we were just discussing this yesterday. We have a technology that to do one tumor costs us $20,000 for one assay. Yeah. Now, we tend to do 5 or 6 assays per tumor, so you can imagine this is now breaking our bank here. And that's why it's so frustrating at a time when we can learn the most if we had the money. The money is, becoming a challenge to us. So that's kind of sad. So we have these amazing tools that allow us to dissect the fundamentals of why breast cancer exists and those proteins that we may target, for example. And then we have these amazing tools. So this is an example of a new technology that came about in the last few years called organoids, where we can now grow breast cancers in the laboratory routinely from any patient. So we've done this about 300 times now on every patient that's come through our clinic, and about 85% of the time we can readily grow their breast cancers in the lab. And the goal is, the classic goal of precision medicine is we should be testing the drug on that thing in the lab and not on you, yeah? We should test all these drugs, find out which one works, and then give it to you, not test on you, which is our current paradigm, yeah? So, very exciting. We can do this all the time, and this, again, will be transformative. There will be, in the next 5 to 10 years, testing like this. Some of you may know there's some companies already doing it. It's pretty early stage, I would say. But we're learning a lot from this. This, I would say, is important in that we run something that's quite hard to talk about, but I would say we run an autopsy program. So many of you are probably organ donors on your driver's license. Once you have advanced breast cancer, you can't donate your organs anymore for obvious reasons, yeah? And so we say to them, well, why you were going to donate anyway on your driver's license, why don't you just donate your organs for research? This is a very challenging thing to posit but a lot of patients, we've signed up 140 patients to do this, and we've done 40 autopsies. We do those at 2 a.m. in the morning. All the volunteers come in. We have a team of 20 that comes in. It's an incredible amount of effort and time and money for us. But we are learning amazing new insights as to why people pass away from breast cancer. This is really important, yeah? The final stage. What is it that causes people to die from breast cancer? And I would just say, this is us growing. We have grown breast cancers. This is breast cancer that's growing in the brain, mesentery, liver, lung, pleural effusion, ascites, spleen, ovary, and omentum. We can now grow these from all over the body and now learn about them. We can have them growing in the lab. We can sequence them. We can test them with drugs. Yeah.
How do I sign up to?
Yeah, I thought you might say that. That's always dangerous when I say this because it's a very good question, and we get this all the time. And we're very passionate about doing this, and we have passionate patients who have signed up and, donating them. And it's a very important, family affair, this whole thing, yeah? But we have to do it fast. So it has to be close to our center. If you don't live close to us, which I'd be sad if you don't, then we can hopefully find another center that can do it. Yeah. So, we're blowing away. But it is a fundamental question, and we hope these autopsy programs are going to increase because there's so much more we can learn now from the autopsy process and the donation of organs. Yeah. Yes. Yeah. Yeah, the tools, it took a long time for the technology companies invested to make the tools, yeah. They tend to start off expensive and they come down over time. But it's a market, it's just market dynamics. And, the tools are incredibly helpful and we're learning a lot. But it's inhibitory to how many we can do. And we need to do a lot because obviously you are all different and all your tumors are different, so we have to do quite a few. I wasn't going to, but I can talk about that. So the question is, what are the advances coming in genetic testing and other targeted agents towards that, such as PARP inhibitors? So one of the advances has been this our ability to sequence. That's come down tremendously. So to sequence a genome used to cost us $1 billion. It now costs us about 400. So that's an example where technologies do get cheaper over time. And so we can sequence all of your tumors now. Many of you probably had your tumors sequenced, and then we can have therapies against it. So I was speaking to a survivor this morning who had had a young lady with triple negative breast cancer. Once she had had triple breast cancer, she was then found out to be BRCA positive, and those tumors can be targeted with the PARP inhibitors. So there are many examples now, ESR1 mutations, PI3 kinase mutations, all of those are coming from our ability to measure them, and that comes from these technology developments that have come through laboratory research in these technology companies. So I think that we're only going to see an increase in these targeted therapies developed to these genetic mutations. I will say one last thing. Am I getting thrown off, or you're okay? I thought I was going to get, like the Oscars, I was going to get moved off the stage. So I will just one last 2 minutes. Artificial intelligence is going to disrupt healthcare. This is going to be a major change, and it's going to be a good change. So how many of you have used ChatGPT or Claude AI? Wow, that's crazy. That's really interesting. I didn't think it would be that much. Nearly everyone. Okay, yeah. So I use it like every morning. This is going to be mostly good. There's going to be, as we move along, there's going to be biases in it. I think you've heard that. There's going to be nefarious people using it. But this is going to allow your physician to really see the bigger picture of you and your tumor that they haven't been able to. So as Isaac said, Facebook captures 200,000 elements of you when you log in. The physician can't possibly, the human brain can take 7 things at one time. And physicians, maybe they get up to 10, but they're not doing more than that. So there's no way they can incorporate everything we now know about a tumor. Just simply isn't. They need support from the computer and generally AI. So this is going to improve our detection of disease. It's going to clearly already improve our drug discovery. There's several drugs entering trials that were developed by AI by itself with no human intervention. It's going to enhance our diagnosis, and it's going to enhance your treatment. Yeah, so it's interesting. I was at a meeting where the CEO of NVIDIA just spoke at a precision medicine meeting. It was a really amazing kind of talk he gave. But I think most people believe there's humans in the loop. That's very rare. Normally there's humans in the loop in these systems. It's very rare that people are thinking about AI alone. But some settings where at some point a human has to look. But I think this is, we are going to collect much more data from you. It's going to be coming through your phones. It's going to be coming through your wearables. The amount of data you see on your iWatch is just like stunning. Yeah, it looks at your step and your gait. These things are going to be good. Yeah, this is going to help us know better about you, your toxicities. This is going to be important for dose reductions, for example. And it's going to be important to knowing how your tumor evolves. I'll just say one last statement that came up about the evolution. So we have a trial. While I said things are going bad in terms of funding, we do have a trial just funded by the government, which is super exciting, and it's an expensive trial. It's called ADAPT. It comes from a group called ARPA-H, which is one of the new institutes of the National Institutes of Health. It's called ADAPT, and it is specifically to look at the evolution of tumors under treatment. So in that trial, we will be doing, with the government's money, multiple assays that are incredibly expensive, working with a bunch of artificial intelligence aficionados and looking at how that tumor is evolving over time and then adapting therapy based upon that change. This is done with the Translational Breast Cancer Research Consortium, the TBCRC, and I think is the next state of the art. We traditionally have been treating just based on one time point, just this is it, and then it doesn't really matter what else happens. But it's the evolution, it's as the disease changes that's most important. So I'm excited about this. I actually do a lot of computer science, so I'm kind of heavily involved in this. And I think this is, while it needs to be moderated, I think this is going to be game-changing. I'd be happy to take any questions on that. Yeah. You're signing up.
Have you done any research with TILs or TILs trials? And if so, can you maybe explain what that is and then tell us all about it? Okay, thanks.
Yes. So I personally haven't because of my PhD, but I can tell you at our institution, yes, TIL therapy is used. So TILs are tumor-infiltrating leukocytes or lymphocytes. So it's where your immune cells have made it into your tumor, but for some reason they're not working. Remember how Isaac said, your tumor's pretty good, the body's pretty good at eliminating this, but there are times when the tumor just evades it. And so these TILs are inactive. So we can take those TILs out, we can re-educate them in the lab as a cellular-based therapy, and then inject them back in. We can boost them, expand them, genetically modify them. We don't do that in breast cancer, but we do it in multiple other tumor types, and I've had some successes with it. So yeah, and hopefully one day we'll be doing that in breast cancer. So yeah, using your immune system, extracting it, putting it back in, that's now going to be, but those cellular therapies are expensive and difficult to do. Yeah. With the hat.
Hi, yeah, somewhat related. So for estrogen positive breast cancer, we've heard a lot that immunotherapy is not responsive. Can you give a little reason behind that? I'm not fully understanding it myself.
Gosh, I wish I knew the reason. We study this. We just published a paper on it actually just recently on why it's so what we call immune cold. It's probably the environment around it that's suppressing those cells from coming in. We call that immune evasion. It has these ways to cause immune evasion. We just don't understand why that's going on. Lots of people trying to study that. There is a cell type called the macrophage that's very prominent in those, and there are targeted therapies against macrophages. So there might be things on the horizon specifically for ER-positive breast cancer, targeting those specifically. Yeah, you always ask the difficult questions. I would say 2 to 3 years. Are we wrapping up, or can I continue on? Okay. Oh, my watch says, okay. Technically, yeah. Okay, then great. Then I will take care, yeah.
Okay, thanks. Do you know about Divitum TKA liquid biopsy test? Okay.
There are lots, I would say, there are lots of tests out there. It's the fastest growing industry in the biomedical industry is liquid biopsy companies. I actually, for my other life, I do 1 day a week in venture capital development in precision medicine, and this is what we see all the time. And this is because it's exciting and everyone's excited about it. But many of them are really young and they're early stage and they're trying to get going. It's best to ask your oncologist or someone, an expert in that field. Yeah, and that's what's happening. I mean, he'll probably say, I don't, or he or she will say, I don't know, because it just got approved, yeah.
Hello
Yes.
again. What's the research that you're doing now that you're super excited about, if it's not a secret?
Well, how about I tell you about my Breastie's funded research?
Okay, I love that.
Yeah, that's good. Actually, that's a great question. I never said that. Yeah. Okay, so talking about foundations that are great for funding research, Breastie's funded, I should say Steffi and my research, because we do that, we're co-partners in this. We found a version of estrogen receptor where it doesn't have this classic mutation like you saw in Serena 6, but it has a different type that's even worse than that. It's something called it's something called a gene fusion, and those are completely resistant to any therapy. And so we are trying to understand the prevalence of that, how often it occurs. We think that occurs in about 1 to 5% of cases of ER-positive advanced breast cancer. And then 2, how do we target it? And I was on a call last night, actually, driving up here, where a company has a pretty exciting way to target those specifically. So again, you're going to ask, well, how soon is that going to be? And I'm going to say, well, it's probably 4 to 5 years. But yeah, no. So Breastie has provided us funds to do that fundamental research. One, how often do we see this? Can we detect it in liquid biopsy? So we developed an assay to do that specifically, and that seems to work. So that's great. And then finally, which is the hardest part, how do we target it? So I'm very excited. We do a lot on liquid biopsies. I personally believe that's going to be the biggest transformation in cancer treatment that we've seen in many years. Yeah, no, exactly. And as I said, we take blood on every advanced breast cancer case at every progression from our clinic. We've done that now 1,500 times and built a large bank, which we think is going to be probably the largest kind of real-world observation of kind of what happens during evolution. Yeah.
Can you speak about CAR T therapies?
So CAR T therapies, very exciting. They work in liquid tumors in the hematology space. So basically, you take your T cell, so we take your immune cells out, engineer them to better find the tumor, and then inject them back in. And they do this through genetic engineering. That worked, for some reason, that worked really well in leukemias, for example, but has worked poorly in solid tumors. And people are trying to understand why it works so poorly in the solid tumors. The challenge is it's extremely expensive. So we've done many of them at our institute. They cost between $1 million to $2 million a time. And obviously we have a health insurance plan as well that's saying, hey, we can't, this is not reimbursable and we can't continue to do this. Yeah. So a lot of research to try and understand, how can we improve their efficacy? And ultimately, these personalized cellular therapies are kind of one of the ways to go, to use boost your own immune system. Yeah.
Can you share with us any kind of resources, information, places that we can learn more about what's happening on an international level, different therapies and research?
That's a great question. I don't think I've ever had that question before. Google, ChatGPT. Yeah, I don't think there is a singular resource for advances. We work a lot with Germany has some great breast cancer groups, some leading ones. France does. Paris has some of the leading groups there, and they all come and we meet them probably every couple of months we're seeing them. But I don't think there's a singular way to get to advances coming out of them. There are meetings over there like ESMO Breast, the European Society for Medical Oncology, the equivalent of ASCO, held its breast meeting in Munich about a month ago, and so most of our people went there. And then ESMO Maine is in September. I'm going there. But I don't think there's a singular way to get to that. Okay.
I was going to say we have time for one more.
And we'll take one more from the hat. You can say your name, and then I don't have to use the hat.
I'm Leanne. I'm also
Leanne.
the hat.
Okay.
This may be a silly question. I don't know. It seems like everything is going down the route of mutation targeting.
Yep.
Can you force a mutation? Can you... just an idea.
Yeah, well, so that's not a silly question. So, for example, when you give chemotherapy, that Isaac talked a lot about, the way that's working, that's damaging DNA and making mutations. And it's making so many that the cancer becomes overwhelmed and then dies. And so one of the challenges is that sometimes people later in life, particularly in children, have developed cancers from that therapy. Yeah, and you can still see what that therapy has done. Yeah, so we don't worry about this in adults. Yeah, but yeah, sometimes. But actually, the evolution of the mutation estrogen receptor is a good example. That thing only appears when you put therapy on. It's a therapy-enriched mutation because you've put pressure on it, and to get around that pressure, it's then expanded that mutation. So it's only in the last 10 years that we have the tools, like I've said, to do this research and understand this. These are you're asking fundamental questions about it, and now we're learning that, and that's how we're going to it's through these liquid biopsies and all of these tools and these models, like I showed, that we're really going to learn about that, and we're going to be using that in 10 years' time. What Isaac said is it's kind of amazing. The main tools we have now are surgery, radiation, and chemotherapy. Yeah, it's kind of weird, that's the same to 50 years ago. So this will change. Okay. Fantastic.
All right, give it up for Dr. Adrian Lee.